Okay, so let's start to pick up where we left off. We ended with damages due to mutagens. So some of those mutagens can be alkylating agents, for example, ethyl methane sulfonate, shown as EMS in this figure. So ethyl methane sulfonate is an alkylating agent. Why? Because it adds an alkyl group to our bases. So in this example, we have guanine that is hydrogen paired with cytosine. Now, in the presence of an alkylating agent like ethyl methane sulfonate, if you will observe, guanine has been alkylated in this side, and as such, this oxygen cannot function in hydrogen bonding anymore. And because this oxygen has been alkylated, the double bond from here has now become a single bond. As such, this bond here forms. So now your nitrogen here does not contain this H anymore. And because of that, it cannot hydrogen bond with cytosine as well. So which means that the effect is thymine becomes the best pair for this alkylated guanine. So if you will find, this technically is a DNA damage. Your base is modified. But if you use this as the reference strand, the complementary strand, thymine, will be incorporated instead of cytosine. As such, a transition has occurred from cytosine to thymine. So in the complementary strand, the sequence has changed. So a mutation has occurred in the complementary strand. Another damage can be caused by UV or ultraviolet. So the usual effect of UV is that if you have two thymines that are adjacent to each other, in the presence of UV, they form pyrimidine dimers, meaning that the two thymines have interacted with each other. So this is one thymine. This is also another thymine. In the presence of UV, they interact with each other and form this ring, this four-membered ring, which we call a cyclobutane ring. So the effect here is that your DNA, so this is one thymine interacting with another thymine, when they bind to each other, a bulge forms in the DNA. Now, this becomes a problem because this bulge cannot be read by the DNA polymerase during replication. As such, the complementary strand once again will have a mutation. So this DNA damage here can also be called a lesion, a DNA lesion. So another mutating agent are free radicals and ionizing radiation. Basically, the both of these involve the use of radicals. For example, your oxyradical. So what is a radical? It's basically something that has a single electron or an unpaired electron. If you have an unpaired electron, it would really want to react with everything else so that it becomes more stable. So because of that, it can cause damage by modifying bases. It can cut single-stranded DNA, or it can cut both strands, so you now have a double strand break, or it can cut between base pairs. So in this example, this is your phosphodiester bond, this is your sugar group, and this is your nitrogenous base, and another phosphodiester bond here. In the presence of an oxyradical, it cleaves or it cuts this part here. so that now you have a broken sugar phosphate backbone. So this is a single strand break basically. Now, if we have several damages that can happen, there are several repair mechanisms that address this. This is an example of a direct repair, meaning if you have a damage, the repair happens exactly on the site of the damage. So for example, you have these two thymines here. In the presence of UV, they have formed a pyrimidine dimer. So the enzyme that usually repairs this in E. coli or in prokaryotes is called DNA photolyase. It's termed photolyase because the photo part means it requires light to activate it. Specifically, photolyases are activated by blue light. So, once the photolyase is activated by blue light, so it's this red circle here, it goes to the site of the damage, and from the term lyase means it breaks apart this this cyclobutane ring that is formed by the UV. So once it breaks the pyrimidine dimer, the thymines go back to their normal structure. Another repair mechanism is using methyltransferase. So from the term transferase, it means that we're transferring one group to the other. So in the example here, we have a modified guanine. It's methylated. And because it's methylated, it cannot hydrogen bond well with cytosine. The enzyme in this example is an O6-methylguanine methyltransferase. The enzyme has a thiol group here, which is SH. So, in the presence of this enzyme, the methyl group is transferred from guanine towards the enzyme. So that now your guanine can hydrogen bond with cytosine, but the enzyme now contains the methyl group. Now, some would say that this enzyme is a fake enzyme, largely because once the methyl group has interacted with the enzyme, this is a permanent bond. So because it's a permanent bond, the enzyme is consumed in the reaction. As such, it's called a suicide enzyme, because now it cannot act as an enzyme anymore. Another direct repair mechanism is using ALK-B, which is an enzyme that makes use of redox reactions to demethylate itself. So the example here is an adenine that is methylated and a cytosine that is methylated. Now, we're not going to go into the detail of the mechanisms, but what I would want you to understand is that ALK-B makes use of redox reactions to remove the methyl groups so that you can have an adenine and a cytosine that is structurally sound. So if we have direct repair, we also have indirect repair. Now, the thing with indirect repair is if you have, let's say, a DNA strand here, and the problem is just in this particular site, with indirect repair, what it does is that it removes the entire fragment of the DNA and patches it up with new DNA. So, it's not like with direct repair wherein the only thing you deal with is the site of the damage. In indirect repair, we remove stretches of DNA and replace it with new DNA. So the first indirect repair mechanism we're studying is nick translation, which is usually done if there is a single strand break in the DNA. So if you will find this gray part here, that means that the sugar phosphate backbone has been damaged. As such, DNA polymerase 1, so this is found in prokaryotes, is recruited so that it can cut up this entire part. Because DNA polymerase 1 is also a polymerase, that means it can cut up this part and also replace it with a new strand. And that new strand is now shown as the red strand here. And so the ending is, this part here now contains the nick. So it's as if the nick has been translated to a new location, except that this nick can be repaired by DNA ligase. So as you can see in this example, we only need polymerase 1 and ligase. In subsequent indirect repair mechanisms, this is quite a common theme in that polymerase is used and ligase is used to seal the nick eventually. So here, in the second indirect repair mechanism, we have mismatch repair, which is usually used if you have a mismatch. So you have here thymine that has been matched to a guanine. So it's a mismatch because guanine is usually paired with cytosine, whereas thymine is usually paired with adenine. Now the problem here is we do not know which is the original strand. Is it the one containing the thymine or is it the one containing the guanine? As such, the parent strand has to be identified. Now, in prokaryotes, usually the parent strand is methylated. The red strand, which is the newly synthesized strand, because it's newly synthesized, it hasn't been methylated yet. As such, we can identify the parent strand or the original sequence. So in this case, there are 3 enzymes that we make use of. We have MutH, which recognizes the parent strand, and MutS, which recognizes the mismatched position, and MutL connects both MutH and MutS to each other so that the strand which contains the wrong base can be removed. So that will be this strand here. So we remove this part and this part. So it's an entire fragment that has been removed by exonuclease 1. So that's the enzyme being used. So once it's removed, DNA polymerase 3 comes in to patch up this part, and DNA ligase comes in to seal the nick. Next is we have base excision repair, which is usually used if you have a damaged base, so a methylated guanine or an alkylated base or a broken base structure. So the first thing you do is DNA glycosylase comes in. So if you will remember, your sugar group is connected to the nitrogenous base via an N-glycosyl bond. So DNA glycosylase attacks this bond here and breaks it so that this base can be removed, leaving an apurinic or apyrimidinic site. Or a more general term would be to call it an abasic site because it does not contain a base. So in this case, an endonuclease comes in. So if you will remember, endo meaning inside and nuclease. So it cuts, so this enzyme cuts the DNA from the inside. So now this endonuclease, once it's cut the sugar phosphate backbone, an end has been generated so that an exonuclease can come in and start to remove the nucleotides. Once the nucleotides have been removed, polymerase comes in to patch up the nicks. And then the remaining nick here is repaired by DNA ligase. The next indirect repair mechanism we're studying is nucleotide excision repair, which is usually done if you have a DNA lesion. So this can be a pyrimidine dimer that has formed in the DNA. In this case, ABCXC nuclease comes in and it cuts this part here and this part here. So this entire fragment has been excised, and DNA polymerase 1 comes in to repair this part, and then ligase comes in to repair the nick in the backbone so that you can have an intact DNA. So far, the examples that we have used have reference strands, meaning we can identify which strand contains the original sequence. However, what do you do if you cannot identify which strand has the original sequence? For example, in this part here, you have an unrepaired lesion. So that means when you start to replicate this strand here, this growing DNA strand stops here because the DNA polymerase cannot read this lesion anymore, which means it wouldn't know what base to incorporate in the complementary strand. So how does an organism circumvent this? So in bacteria, for example, they can either go for recombinational DNA repair or for error-prone repair, which will be discussed more in the next slide. On the other hand, if you have an unrepaired break here, for example, if you try to replicate that part, because this was a break, the effect is the complementary strand wouldn't know how to push forward as it has no reference strand anymore. So the effect is you have a double-stranded break here. So what does the organism do? It goes for recombinational DNA repair, which will be further discussed in the next how many slides. So, SOS response is also known as the error-prone response. Now, or error-prone repair. We say that it is error-prone because, for example, here we have this strand which contains the lesion, this part. The DNA polymerase cannot read this part, and as such, it wouldn't know what nucleotide to incorporate. So the effect is that DNA polymerase guesses and randomly puts in whatever nucleotide it can put here until it encounters the reference strand and just copy that reference strand faithfully. So this is kind of like when you have when you encounter a multiple choice question and you don't know how to answer it. So if you don't know how to answer it, you just randomly guess A, B, C, or D. So you'd rather have an answer than to leave the question blank. So that's how error-prone repair also works. The DNA polymerase would rather have some random bases placed here than to leave it blank and to terminate replication prematurely. So because of that, this part here can be full of bases that are not reflective of the reference strand or are all wrong. But the advantage is at least you heighten the chances of the bacteria to survive. So that somehow if you get past this point, you once again encounter the reference strand and just copy it the way it is. Now, what if you have a double strand break? If you have a double strand break, recombinational repair is needed. So we're not going to discuss in detail the mechanism of recombinational repair, but the point here is, if you have a double strand break here, it is joined to another DNA strand, and you will be hoping that this repair mechanism is the real sequence of the DNA. So you're basically trying to take a risk with this. So the effect is this part here is joined to this part. There you go. And then these flaps are eventually removed so that you have one intact DNA, which hopefully contains the correct sequence. Hopefully. So this micrograph here shows shows the recombinational repair happening in this part. So if you will remember in our discussion on DNA replication, we said that the ends of the DNA have to be tucked in so that repair enzymes will not touch them, basically. So it's because if you just have free ends like these, they are assumed to be double strand breaks, and so recombinational repair might ensue. That's why in replication, we tuck the ends in. And so this is the end of our discussion on mutation and repair. So I will be showing a video on sickle cell anemia to highlight the importance of studying mutation and repair.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under low oxygen conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need.
So if you see this part, this looks like a sickle. So the sickle is yung parang yung hook ni kamatayan, parang ganon. So yun, that's the sickle.
Sickle cell anemia is caused by a single letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in a position where glutamic acid should be. The valine makes the hemoglobin molecules stick together when oxygen tension is low, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
So if you will see, based on that video, it's a single change in DNA that has caused the change in the structure of the red blood cell, causing sickle cell anemia. So please make sure that you read on repair and mutations in your module or in any biochemistry book, and to prepare questions if you have any for class.